Hello everyone. Let's start our S R Kantwa objective series. That is the fourth chapter, genetics in plant breeding. Let's start with our first question. An organism which has acquired a heritable variation as a result of a sudden change in the hereditary material. It is called as in the mutant. Question number second. A polyploid having the chromosome set from the different sources, such as the different species, is known as the allopolyploid. The sex form in most of the cucurbits, including the melons, is monoecious. The increased performance of the F1 hybrid over its parent is called as an heterosis. The ratio of the genetic variance to the total variance, which determines the relationship between the phenotype and the genotype, is known as its heritability. Question number 6. The mass pedigree selection method of breeding is particularly useful for the improvement of that is option A, self-pollinated crops. Question number 7. Emasculation of a flower is necessary for the hybridization in option B, self-pollinate crops. Question number 8. Leafy vegetables like palak, amaranthus, spinach are highly cross-pollinated. Question number 9. The modern tea roses were involved as a result of which of the following crossing? Of the crossing of the floribunda into tea roses. Question number 10. Mendel did not face the problem of the linkage. However, the seven characters started by him were located on how many chromosomes? That is 4. The test cross ratio in the case of the duplicate gene action will be 3 is to 1. Yes. Question number 12. 80 ratio in one strand of the DNA duplex is 2. What will be the ratio in the complementary strand? It will be option B, 0.5. Question number 13. The term gene was coined by Johnson. Question number 14. The number of chromosome in wheat endosperm is, yes, here is asking it's an endosperm. It will be 63. Question number 15. The cytoplasmic male sterility is best suited for the hybrid seed production in option third, vegetatively propagated crops. Question number 16. The multiple effects of the single gene is known as a pleiotropy. Question number 17. An example of the heterozygous but homozygous population is, it is option C, hybrid variety. Question number 18. The cross of an inbreed line with an open pollinated variety is known as the top cross. Question number 19. The artificial hybrid was made by, yes, Thomas Fairchild. It was a cross between carnation and the sweet William. Question number 20. The synthetic hybridization on a scientific basis began with the work of option C, Coleodoter nicotiana. Question number 21. Double cross plan was suggested by, yes, it was suggested by Jones. In maize he has done the double cross. It was started in by D.F. Jones in maize. Question number 22. Who gave the concept of the circular chromosome in the E. coli? It was given by Cadians. Option A. Question number 23. Sex in plants was discovered by, it was discovered by option B, Chameleons. Question number 24. Who produced puppies through the artificial insemination in dogs? It was done by option C. Splanjaning. Question number 25. Yes, the concept of epigenesis was proposed by Wolf. That is option C. Yes. Question number 26. The origin of the species was published in B, that is 1859. Germplasm theory was proposed by Weizmann. Cell linkage theory was proposed by, yes, that is Virchow. Question number 29. The chromosomes are the most condensed in which stages? In anaphase stage of the, yes. Question number 13. The DNA synthesis takes place in which stage? It takes in the S phase. Question number 31. In the DNA double helix, that is G by A is equal to the, it's equal to the, that is option A, C by T. Question number 32. Hershey and Chase in the experiments labeled the DNA with, yes, DNA was labeled with P32, that is option 3. Question number 33. The number of bases in the single turn of the DNA helix is, it is option third 10. Question number 34. The common form of DNA present in the living organism, yes, it's a B form. Question number 35. The DNA polymerase first was discovered by, yes, the DNA polymerase first was discovered by the Kornberg, option third. Question number 36. The DNA replicating enzyme in bacteria is option C, DNA polymerase third. Question number 37. The proteins involved in keeping the DNA single stranded during the replication is, it was option D, SSB protein. Question number 38. The Okazaki fragments are formed due to the synthesis of DNA in the direction, yes, it was in the direction of 5 dash to 3 dash. Question number 39. Deamination of the adenine leads to the formation of the, option B, hypoxanthine. Question number 40. The maximum absorbance of the DNA molecule is at dash OA wavelength, at 206 wavelength. Question number 41. The Drosophila 16A region of the X chromosome causes normal eye. If the region is repleted, the eye produced is, its option C, bar. Question number 42. The resulting two daughter cells produced by the bridge breakage fusion will have both, that is both A and B, duplication and the deficiency. Question number 43. A tautomeric shift in the adenine, that is amino to immuno, allows it to pair with dash only, with the cytosine only. Question number 44. In the humans, an XO individual has how many bar body in her cells? It has none. Question number 45. That is, the nucleolus plays an important role in the production of, yes, nucleolus plays an important role in the production of the rRNA. Question number 46. An example of the dicotyledonous albuminous seed is, it is soya bean. Yes. Dicotyledonous albuminous seed is soya bean. Question number 47. An amino acid which is related to the drought tolerance in plant is, It is option B, proline. Yes. Which ammonia acid produced during the drought? It is the proline. Let's move to question number 48. Which of the following are the inhibitors of the cell synthesis? It's option D, all. That is penicillin, oxamycin, rictocatine, and then vancomycin. Question number 49. Inhibitors of the cell membrane synthesis are, it's option D, all of these. That is polymyxine, gramicidine, and the circulin, and triosidine. Question number 50. Inhibitors to the mRNA synthesis transcription are, it's option D, that is all. First, actinomycin D, then B, rifamycin, and third, fibromycin. Yes, question number 51. The inhibitors to translocation, that is protein synthesis, are, it's again option D, all of the above, that is streptomycin, euromycin, and neomycin, and chloram, chloramfenol and erythromycin. Question number 52. What is the seed multiplication ratio in the chickpea and wheat? Are respectively, it is option D, 1 is to 10 and 1 is to 20. Question number 53. What is the seed multiplication ratio in the potato and the groundnut respectively? It is option A, 1 is to 4 and 1 is to 8. Question number 54. The seed multiplication ratio in the paddy is, it is option B, 1 is to 80. Seed multiplication ratio in the bajra is, it is again option C, 100, 1 is to 200. Seed multiplication ratio of the rapeseed, red gram, hybrid maize, sorghum, and jute is, it is option A, that is 1 is to 100. Green plant photosynthesis takes place in, it takes place in option D, chloroplast. Question number 58. The site of the protein synthesis of the cell is, that is ribosome. Which cell organelle is having a diagnostic enzyme? It is the digestive enzyme, lysozymes. Question number 60. Who discovered the X-rays? It was discovered by, yes, it was discovered by the Roentgen. 61. The defective copies of the normal are known as the pseudogene. Normal gene is known as the pseudogene. Question number 62. The term the term the term, this is muton and cistron, coined by 1957 by Benzer. The variance which is fixable is additive. Question number 64. The term hybrid refers to the somatic hybrids. Now question number 65. Which component of the immune response is crippled in the AIDS? It is in helper T cell. Question number 66. During the mitosis, chromosomal DNA duplication takes place in the interphase. The cell wall of the fungi are made up of the chitin. Question number 68. Which of the following compound contains the cobalt? Which of the following compounds? Yes, it is vitamin B12. Option B. Question number 69. Antibiotic that causes the misincorporation of the amino acid and the protein synthesis is puromycin. That is option C. Question number 70. DNA as a genetic element was shown by the experiment of, Yes, it's option A. Avery, McClurdy, and McCarthy. Question number 71. The inherent capacity of an organism to increase in numbers under the ideal condition is known as the reproductive potential. Question number 72. rDNA analysis provides information on the phylogenetic relationship. Now, question number 73. The wetlands are threatened due to, it is threatened due to the option C, silting due to erosion. Let's move to question number 74. Cryopreservation is not suitable for preserving organisms which are that is option C, aerobic. Question number 75. The Tiger Project is a holistic approach for the conservation of the ecosystem. Question number 76. The two species belonging to the same genus are identical in the morphology. Which of the following structural characteristics is more useful in distinguishing the such species? It is option C, chromosome number and morphology. Question number 77. The two taxonomic species are distinguished from each other by their failure to the inbreeding. Question number 78. A common crop which is grown in the natural autoploidy grown. Yes, it is potato. It is the that is 48 chromosome number. Question number 79. The pearl millet is predominantly cross pollinated because it is protogynous. Question number 80. A F1 plant with the genetic constitution capital A, capital B, small c, capital D, and small d is a test cross with all that is small A, B, B, C, C, D, D. There will be yes 8 genotypes. Question number 81. In wheat and the rice, the genetic purity of the variety deteriorate due to that is option C, mechanical contamination with the seeds of the other varieties. Question number 82. A mating among the following cannot lead to the heterosis. That is option C. Yes, that is capital A, capital A, capital B, small c, small d into capital A, capital B, small d, small d. Question number 83. What is the most useful method of detecting the virus infection in the seeds? Yes, it is ELISA. The most important factor which determines the stability of the seed is, yes, it is that is option third, moisture. Question number 85. In seed testing, the grow out test is conducted to determine the genetic purity. Yes. Question number 85. Translocation of the carbohydrate mostly takes place in the form of, yes, it takes place in the form of the that is sucrose. Question number 87. If the gametic composition of the genotype is capital A, small b, capital C, that is ABC, and ABC, in equal proportion, the genotype of the source individual will be capital A, small b, capital C, small c. Question number 88. The stalk of the ovule is called, yes, it is called option C, funicle. Question number 89. IB, IPGRI headquarters is situated in, it is in Italy, Rome, Italy. Question number 90. A fertilized mature ovule which possesses an embryonic plant food reserved and a protective coating in a plane in a species is in a plant species that is the seed. Question number it's not visible much. Question number 91, I guess. Yes. The visible scar observed on the seed at the piece of the detachment from the seed stalk is known as that is option 91, right? Yes, option D, helium. 92. What is this? Aflatoxin are produced by it is produced by sea fungi. Question number 93. Golgi apparatus is also known as it's option C, dioctosome. Question number 94. The law of homologous series is given by yes, it is given by the Babylon. Yes. Question number 95. Transposable elements were discovered by yes, McClintock. Question number 96. Bubble hypothesis is given by Crick. Question number 97. Yes, gene for gene hypothesis is proposed by that is option C, floor. Yes, question number 98. The pure line variety is a yes, it is a homozygous and homogeneous. Very well know we know this. Yes. Question number 99. Land races are that is option C. They are the local varieties. Yes. Question number 100. Lysine is a limiting amino acid and it is a in the cereals. Yes. Question number 101. In pulses, the limiting amino acids are that is option D, both A and B, that is methionine and then tryptophan. Question number 102. The low neurotoxin variety of the Lithyrus is option C, that is Pusha 24. Question number 103. Pusa Sugandha 5 is a new variety of rice. Yes. Saktiman. Question number 104. Saktiman is a new variety of option C, maize. Question number 105. Pusa Gold is a new variety of wheat. Question number 106. The Pusa 1088, Pusa 1103, and Pusha 1105 are the new varieties of the option A. It is of the chickpea. Question number 107. Pusha Charisma and Mahak are the new varieties of option C, mustard. Yes. Question number 108. Pusa Arunima and Pusha Surya are the new varieties of mango. Yes. Question number 109. Purva, Urja and Pusha Visesh are the new varieties of option B, wheat. Question number 110. Pusa Ratna is a new variety of the option D, mung bean. Yes, Ratna. 111. The source of the dwarfing gene in wheat is option B, Norrin 10. Question number 112. Dgbogen is the source of the dwarfing gene in and it is in rice. Question number 113. The ratio of the economic yield to the biological yield, it is known as the harvest index. Yes. Question number 114. The plant type concept was first introduced by, it was introduced by the, yes, Jennings in 1964. Question number 115. There is an increase in dash under the drought condition. Yes, I know. It is of the all of these. ABA content also is also increased. Ethylene level is also, and then proline content is also increased in drought condition. Yes. Question number 116. The concept of the vertical and this horizontal resistance was introduced by, yes, I know this, it is of the Vander Planck in 1963. Question number 117. The concept of the physiological races was given by, it was actually given by A. Barros in 1911. Question number 118. The source of the A genome in wheat is, it is option A. Triticum uratum. Question number 119. An amphi diploid between the Brassica nigra and Brassica campestris is, that is option C. Yes, Brassica juncea. Question number 120. What made the first intergeneric cross between the bred wheat and the ray? I know it's option B, yes, Rimpo in 1928. Question number 121. The BTT is associated with, it is associated with, yes, it is associated with the option D, barley. Triploids are normally sterile. Question number 123. The first man made a cereal is, absolutely, it's critical. Question number 124. The cells without cell wall are known as, it's a protoplast. Question number 125. The condition in which the F1 plants interspecific crosses are vigorous, fertile, but the F2 progeny is weak and sterile. Oh, it is option C, hybrid breakdown. Yes. Now, question number 126. The inability of a hybrid to produce viable offspring is known as the hybrid sterility. Yes. Question number 127. The term heterosis was given by Piano by Schull in 1914. In question number 128, the highest uniformity is present in, it is a present in, that is option C, single cross. Yes, double top cross refers to, that is option A into B into OPV. I guess it's written that only. Okay, so question number 130. The dominance hypothesis of hysterosis was first proposed by, it was proposed by Davenport in 1908. Question number 131. The method used for the fixation of the heterosis is, yes, it is, that is option D, all of these. That is apomixis, polyploidy, and asexual reproduction. Question number 132. In a cross-pollinated species, a true breeding line obtained by the continuous inbreeding is called as, it is called as option B, inbreed. Question number 133. The progeny of the single plant obtained by the asexual reproduction is, yes, we all know it's clone. Yes. The progeny of a single homozygous self-pollinated plant is known as, it is known as the pure line. Yes. Question number 135. The pure line theory was given by Johnson. Question 136. The term genetics is coined by, it is coined by Batson. Question number 137. The phenomenon of linkage was first observed by, it was absolutely observed by Batson and Punnett. Question number 138. One gene, one enzyme hypothesis was given by, Yes, it was given by Beadle and then Tatum. Question 139. The Puran concept of the gene regulation was proposed by Jacob Monod. Question number 140. Dr. Hargobind Khurana was awarded the Nobel Prize, yes, in 1968 for his work on, yes, that is option C, genetic code and in vitro synthesis of the DNA. Question number 141. Which of the following sets is the correct sequence of the events in the cell? Yes, first it is transcription, then it is translation, then it is protein synthesis. What's this transcription? It is from the DNA to RNA. Translation is from RNA to protein, and then it is the protein synthesis. Now, question number 142. The performance of the double cross hybrid, yes, A into B, then into B into D, can be predicted from the average performance of the combination of, that is option B, AB, AD, BC, and CD. Question number 143. The number of the single crosses will be equals to, it will be equals to, that is option A, n bracket n minus 1 divided by 2. Question number 144. The number of the double crosses will be, yes, it is will be option B, n bracket n minus 1 bracket n minus 2 bracket n minus 3 divided by 8. Question number 145. The number of three-way crosses are, yes, it's option A, n bracket n minus 1 bracket close, then again bracket n minus 2 divided by this 2. Yes. Now question number 143. The variety of the wheat developed from the spring into wheat winter wheat crosses is termed as an, it is termed as an option D, that is, no, it's like VEEBY. Question number 147. The intermediate effect of the pollen on the character of the endosperm is called as an, it is called as an option B, that is genia. Yes. Question number 148. The individual with the gametic chromosome number is, yes, it is all, yeah, we all know it is haploid. Question number 149. The individuals with the second chromosome are commonly known as, it is known as, that is option B, aneuploidy. Question number 150. Pollination and the fertilization is an unopened flower bud. Is the situation called as an, yes, we all know it is called as in the cleistogamy. Question number 151. The situation in which the one gene, yes, what is this, hide the effect of the second gene when both are present on the chromosome. It is called as an epistasis. Question number 152. An individual with the yes 2 and minus 1 chromosome constitutes, it is an, yes, I know, it is an option B, double monosomic. Question 153. Which of the following statement regarding selfing is correct? It is, that is option C, reduces the heterozygosity. Question number 154. Triticale is an, it is an intergeneric cross. Question number 155. The cabbage is a cross pollinated owing to, because it is self incompatibility. In 156 in maize, the cross pollination occurs due to, it occurs due to the potandry. Yes. Now question number 157. Hello everyone. So let's continue our S R Kantwa objective questions of plant breeding and genetics in plant breeding. Till question number 1 to over 56 we have done. So let's continue with this 257. Okay. So question number 157. Yes. So question number 157. The bulk population breeding in us is suitable for. It is suitable for. That is option C. The small grain crops. Question number 158. In sugarcane breeding, the initial selection after hybridization is done in the generation. It is done in the generation. That is option B. F1 generation. 159. In a synthetic variety of maize produced by combining 10 lines, the loss of the excess vigor in F2 generation will be, that is option D, 1 by 10th. Question number 160. The cluberium in the random mating population is disturbed by selection, mutation, breeding, and migration. And the genetic drift in which the situation the population may regain its original composition. It is that is option B, mutation. Now consider the following genotypes: breeding lines, second land race, third variety under cultivation. What is the correct sequence of these in terms of the importance to the farmers? It is that is option B, 2, 3, 1. So first is the land race, then it's variety under cultivation, and then it is the breeding lines. Now question number 162. The smallest unit of the genetic material that cannot be divided but can be changeable among the units refers to the option C, recon. Question number 163. What is the possible reason for the ability of most of the tomato varieties in the market to be almost uniformly oval shaped and firm? It is that option B, reduction in the genetic diversity. Question number 164. It is in a cross if we get 8 types of gametes, both male and female parents, from both male and female parents, the resultant selfing will produce a ratio of about, it will produce 27 is to 9 is to 9 is to 3 is to 3 is to 1. That is question option C. Question number 165. The seedlesses and watermelon is a triploid. Question number 166. Which of the following rice variety has been developed through the induced mutagenesis? It is by the option D, Jagannath. Question number 167. In back cross breeding, repeated crossing with the recurrent parent is done to. It is done to option number D, that is completely recover the genotypes of the recurrent parent. Question number 168. In apomixis, the progeny closely resembles the female parent. Now question number 169. The superiority of the F1 hybrid over the standard complete commercial variety is called as the economic heterosis. Question number 170. The inbred lines in maize are maintained by, it is maintained by option A, that is self-pollination. Question number 171. The NBPGI is located at, it is absolutely located at New Delhi. Question number 172. The component lines used in the development of the composite variety of maize are, that is D, open pollinated varieties. Question number 173. Mutation due to change in a gene is because of the, it is because of the, that is D, all of these, that is loss, degeneration, and addition. Question number 174. The chemical mutagens usually induce, that is B, gene mutation. Question number 175. This hybrid is, it is a hybrid formed by the cell division. Question number 176. The reversion occurs due to, it occurs due to, that is mutation. The breeding method for the transfer of the monogenic disease resistance from species to the improved variety of the wheat is, It is that is option third, back across method. 178. The physical basis of the life is, it is the protoplasm. Yes. Question number 179. Botanically, pineapple fruit is a, it is a Soroses. The first hybrid in pulse, that is Arhar, in India developed whose genetic male sterility is, it is no, it is B, ICPH. That is the yes, one ICPH8. Question number 181. The first cotton hybrid variety in the world developed in India. India, the variety is, yes, I see that is option A, H4. Yes, this is the cotton F1 first hybrid. 182. Dr. C.R. Rao is a famous plant breeder working on, yes, he has been working on the sorghum. In which crop the first transgenic was developed? It was a developed, the first transgenic it was developed in this tobacco. In 184, the flavored savor is associated with, absolutely associated with the tomato. Okay. Question number 185. The capacity of a cell to develop into a complete plant, that is the totipotency. Question number 186. That is the bulbosum technique is used in a barley for the production of, that is option B, haploids. Question number 187. Wheat is naturally, it is naturally the segmental allopolyploids. Question number 188. The colchicine refers interferes with, it absolutely interferes with the formation of stem spindle fibers. Question number 189. The colchicine is a generally used form. It is used form, that is option A, chromosome doubling. 190. The sum total of the detritus gene present in the species is known as, it is actually known as, yes, it is known as the genetic load. Chromosome number one, question number 191. The chromosomal theory of inheritance. Yes, it was given by the, it was given by the Sutton and Body. Question number 192. The multiple factor hypothesis is given by, it was actually given by, that is option B, Nelson L. Question number 113, 193. Reverse transcription is also known as, it is actually known as the feminism. 194. RFLP is a, it is a molecular marker. Question number 195. Who is known as the father of the genetics? It is absolutely Mendel. Question number 196. Double helical structure of the DNA was shown by, it was shown by the Watson and Crick. Question number 197. The F2 ratio in a complementary action is, that is option B, 9:7. In 198, the test cross ratio in the complementary action is, yes, it is absolutely 1:3. Question 199. The F2 ratio in a polymeric gene action is, it is option C, 9:6:1. Question number 200. Translocation involves, it involves the homozygous chromosome. Question number 201. Chromosome other than the sex chromosome are called as in, they are called as in autosomes. Then question number 202. An exchange of genetic material between the homologous chromosome is known as the absolutely crossing over. Question number 203. The word mutation was first used by, it was used by Euclides. Coupling and repulsion hypothesis was given by, it was, I guess it was given by, it was given by Watson. Question number 205. The theory of gametes was proposed by, it was proposed by Darwin. Question number 206. The incompatibility is very common in the plant kingdom was reported by, it was reported by Lewis. That is option B. Question number 207. The improved seed distributed to the farmer for the commercial cultivation is, that is option D, certified one. Question number 208. D genome of the common cultivated wheat has been contributed by, it has been contributed by option D, that is the A squamosome. Question number 209. The recommended concentration of the EMS for the induced metagenesis is, that is option A, 0.03, I guess, to 1.15. Question number 10. When chiasma frequency is 100%, the percentage of the crossing over is, it is, I think it's D, 25%. Question number 211. The tissue which nourishes the microspore is, yes, it is, that is the tapetum. Nourishes the microspore. I think it should be, yes, tapetum. Okay. Question number 212. The analog of the fibromycin is, yes, it is, that is option A, thymine. Question number 213. How many the mitotic division will take place to produce 512 cells for the single parent cell? It will be, that is option D, 511. Question number 114. The cDNA libraries can be separated by isolating mRNA. Question number 215. The substitution haploids was reported by n minus 1 plus 1. How many phenotypes are possible in the ABC blood group? In the, that is option B, 4. Question number 217. What is this? From the, no, but when the two heterozygotes for the albinism, what is the chance that the first would be the non-albino? That is question number 217. It is 3 by 4th. Question number 218. DNA can be denatured, yes, by pairing at a temperature of, that is the, that is option D, 94 degrees Celsius. Question number 200 and, sorry, 218 is this. 219. The following amongst the molecule markers and the codominant in nature is, that is option B, SSR and RFLP. Question number 220. The genomic imprinting occurs in, that is, it occurs in, yes, it occurs in, that is option D, all of these plants, animals, and humans. Question number 221. If the DNA of a species has the mole fraction of G plus C is equals to 0.48, the mole fraction A will be, that is option B, 0.26. 222. Which of the following is not a protein? Yes, which is the following? That is the C, ribozyme. Yes, it is not a protein. Virus free clone from a virus infected plants can be obtained by absolutism stem culture. Question number 224. Self splicing introns are found in, they are found in option B, rRNA genes. Question number 225. Oxosome, yes, occurs in, that is option D, mitochondria. 216. The mitosis longest phase is, that is option C, prophase. Question number 227. The nullisomic condition is represented by, that is option C, 2n minus 2. 218. Who discovered the triangular of this brassica? That is option B, Negaheru. Yes. Question number 219. The combination of the male and female features in a body of an individual is, that is known as the sex 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 mosaic. Question number 213. The collection of the germplasm from the foreign countries is called as an, yes, it is called as, and that is option C, the exotic collection. Question number 231. The best method for developing the disease resistance plant is, that is option C, the backcrossing. Question number 232. The growth of the tissue of the living plants on a suitable culture media is called as an plant tissue culture. Question number 233. Clonal selection is used in, it is actually used in, that is option B, the non-flowering species. Question number 234. Population which intermate freely are called as an, it is called as an, that is option D, all of these, that is random mating population, Mendelian population, and then panmetetic population. Okay, so question number 235. Genetically most pure seed is that of the nucleus seed. Yes. Question number 236. Which of the following event is given in are the correct? That is option A. Duration of the prophase is larger than the anaphase. Question number 227. What is the most effective method for transfer of an oligogenic character in an otherwise agronomically superior genotype? That is option D, backcross. Question number 238. Maintenance and multiplication of a gene line used in a crop like pearl millet is done through, that is option A, crossing the plants of A line with those of the B line. Question number 239. Cross pollination in maize is mainly on account of which of the following? That is option B, monoism. Question number 240. Gene for gene hypothesis regarding the disease resistance in the crop varieties have the features of, that is option C, both A and B. And what are those? Those are, that is, this hypothesis was proposed by the rust and linseed by the flow. Yes, for each gene, for each resistance in the host plant, there is a corresponding gene for the virulence in the pathogen. Yes, both are correct. Question number 241. Which of the following statement given below are correct? That is, it is option C, both A and B. That is, additive genetic variants can utilize to develop the variety, and the dominance variants can be utilized for developing the hybrids. Question number 242. Which of the following is the simplest method for testing the gene combining ability of an inbreed lines? That is option that is option C, top cross. Question number 243. Consider the following landmark evolved in the crop variety developed. That is option B, 231. First will we have the semi-top varieties of the wheat, and then we have the hybrid cotton and development of the transgenic crop. Question number 224. Which system is used for the hybrid production in onion? That is C, cytoplasmic male sterility. Question number 245. Which of the following breeding method is based on the competitiveness of the genotype under the adverse environmental condition? That is option B, the bulk method. Question number 246. Uh, mm, a commercially cultivated plant species has the hybrid chromosome number, that is 2n is equals to 48. So what will a plant develop from the pollen culture of these species be? That is the option B, dihaploid. Question number 247. Which of the following mutagens is a non-ionizing? Absolutely, it's UV rays. Question number 242. Which of the following pairs is not correctly matched? It is, that is option D, pearl millet, proto and protanders. Question number 249. The factor responsible for the cytoplasmic male sterility in the plants are located in the, they are located in option D, mitochondrial DNA. Question number 250. Which of the following is not correctly matched? That is option number B, Triticum aestivum. 24. Yes, it is actually this 42, I guess. Question number 251. Which of the following is not correctly matched? It is that is option D, duplicate gene. 14 is to 2. No, it's wrong. Yes, it is that is 15 is to 1. Question number 252. The phenotype P is the function of the genotype and the environment, but when an individual genotype is subject to the multi-environmental testing, the genotypic expression is the result of the option D, G plus E plus divided by a bracket of G multiplication with the E. Match the following question 253 bidding objective with the list to bidding method and select the correct answer using the code given below. Okay, so what it would be? It would be that is option D 3452. So A goes with 3, that is addition of a single resistant gene into an adopted variety, that is the backcross. Okay, then second one, combining useful traits of the two different varieties together, that is option 4. That is what is mutation doing over here? Okay, that is for recombinant breeding. Then question 3, that is creation of the new variability, it is mutation. Option D, exploitation of the non-additive genetic variants, that is second one, that is the heterosis. Okay. Question number 254. The male gametic nuclei are the result of the mitotic division. And from which of the following, that is the genetic nucleus. Question number 255. When both the alleles of a gene are fully expressed in heterozygote, what is this phenomenon called? It is called as in codominance. Question number 256. Which of the following cell organelle causes the male sterility in mustard? That is mitochondrion only. Question number 257. Yes, this is the last one. The center of diversity refers to an area where the cultivated plant species and their wild relatives show. That is option C, both A and B, highest variation and then highest ecosystem. Okay, this is all from my part. Thank you to all of you.